All right. So for those of you who've joined the past few minutes, thank you again for showing up. I really, really, really encourage you guys to just, if you know anybody else who might benefit from joining this study group, I mean, it's free. So pretty easy, just kind of have them go onto this website or shoot me a message with their email and I'll add them to the group. That way, if they can't make it, I got another one. Hey, Sandra. That way, if they can't make it, they can always get the recordings right afterwards. So I'm just going to put a quick reminder for those of you who are not on the call yet to please join the call by dialing the number on the screen. For the best audio, because otherwise it's not so clear when you use your computer. So last time I got some messages from people saying that they wanted to do a biological science passage, especially because some of them are jumping on to the AAMC section bank passages. And so I will definitely be doing a biological science passage today, unless if anybody has any suggestions, you can definitely type that into the chat right here. So suggest a passage here. Or if you have any questions, anything you want me to stop, like if you want me to stop during the presentation, you can always say stop. You can say speed up, slow down. This is, again, meant for you guys. It's not really meant for me. So the more engaged you guys are, the more questions you write down, or the more times you kind of call for my attention, the more interactive and valuable it can be for you. So don't be shy. Any questions are great. And so we're just going to go ahead and briefly overview, while you guys think about a passage that maybe you want to do, we'll go ahead and briefly overview what this presentation will be. Again, I always like to start with these two lines right here. And these two lines, basically, when I think about I interview almost every student that I've used to tutor, and every time I would ask for the two most important things that they needed to succeed. Essentially, it boiled down to these two things, and one was having a process to study. And what I mean by that is, a lot of times, I know especially for me when I first started studying, it was very haphazard. I would be going on to Student Doctor Network, or I'd be going on to Reddit, or I'd be going on to YouTube, or just I think it's like Yahoo Answers, and I would be trying to just get like the best, like what do I study, what do I do, which books do I buy? It's all this confusing knowledge and misinformation that I would be getting. It would be very, very difficult to synthesize it into an action plan so that I know exactly what do I need to do every single day. When I'm alone, when I have to commit 8 hours a day to studying, what do I do? That's the first thing. So people who are successful seem to have that process down, and they can tell it to you in their sleep. The second thing is the motivation to commit. So yeah, okay, so let's say that you got a process to study now. Well, you got to actually commit to it. You got to actually be able to stay in your room or the library, put your head down and study. But the only way that you can do that is if you have a very clear vision, you have a very clear motivation as to why you will be studying. And a term from the behavioral sciences section that you guys should know is this thing behind the locus of control. Now, a locus of control, just, again, bear with me for a second, but you have internal and you have external. A person who has an external locus of control will fail an exam and say that the professor grades too harshly. A person who has an internal locus of control will fail an exam and say, I didn't study hard enough. So it seems to be that the people who have internal locus of controls have more intrinsic motivation, have more capacity to get through the hundreds of hours that it takes. So finding something internally that can drive you seems to be a more successful motivator. Now, in my case, for me, I had taken the test 3 times, and so the third time that I was studying for it, I didn't even think about medical school, honestly. It wasn't even about like becoming a doctor or helping people or saving people's lives or any of that stuff. For me, it was simply I didn't even have the option to go to medical school. On paper, I wasn't even good enough, even though in my heart I knew I was. And so I just wanted freedom from this exam. I wanted to be able to prove to myself that I was good enough and that I could give myself the option, because let's say I didn't go to medical school, I definitely did not want that path to be closed because I didn't do well on the test. So that was my motivation to commit. And so I had the process and I had the motivation, and I was able to improve my score to the point where I was satisfied. Now, the process that I use, it's a little silly, I know, but I call it the boogeyman method, because when I was first trying to explain to people, why this process worked, I was trying to tell them that, like, I was terrified of the MCAT. Like, it was really, really scary. But then the reason why I figured that out that it was scary was because a lot of it was just in my mind. I was telling myself these scary stories about the MCAT, just like the boogeyman. And, with the boogeyman, when you're growing up, right, again, just to tell this quick story, you're afraid of many things. When you're in your room, sometimes you see a shadow and you get spooked, and then your parents come in and they turn the lights on and say, "Don't worry, it's just a teddy bear in the corner," or it's just a tree branch tapping on your window. And so you're no longer afraid of that stuff because you've understood what it is, and you understood that a lot of it was just to own stories that you would make up in your head. And this process allowed me to systematically pick apart the MCAT and extract every single detail that I didn't know, and then I would study it until I knew it. So it's a method to help you shine a light on the MCAT, and that's why I call it that. But we'll go through that today as well. Now, I'm just going to quickly mute. That was not what I wanted to do. 1 second. There we go. Okay. I've identified where the noise was coming from. Dave, sorry about that. I had to mute you, bud, but there was a lot of static. Just type in when you want to speak, and then I'll unmute you. Okay. So back to the presentation. Now, we've talked about the 2 most important things, in my opinion, that you need to succeed in the MCAT, and that's to have a process to study and the motivation to commit. I just recapped the boogeyman method. Essentially, it's about practicing a passage, analyzing the passage, organizing the passage, and then drilling the passage. And so, any suggestions? Have you guys gotten a chance to think about anything you'd like to go over? Otherwise, we're going to go over a biological science passage. Feel free to type it in the chat. Okay, so we will go over that then. Now, let's quickly do this. So, in order for this, in my opinion, lesson, I know some of you have seen this passage before, but it's still important to go over the strategy. Now, what I want to do is, I'm going to briefly describe to you the approach that I use for all the science passages, and then I want to walk through the passage with you. So let's jump right in. When I'm approaching a science passage, the first thing I do is, if there's a title, I really like to read the title. And the reason why, if anybody can think about it, is due to priming. So whenever I read the title, it's almost like you're telling your brain, okay, reach into this file cabinet and start to pull out only the relevant files. For example, if this is a passage about biochemistry, Okay, brain, start to only pull out the biochemistry files now so that I can get more and more primed for what's to come. So if we read the title, it says adapted from Kawaguchi, and the title is A Membrane Receptor for Retinol Binding Protein Mediates Cellular Uptake of Vitamin A. Okay, so from this title alone, we can get a pretty good impression of what the passage is going to be. So it's about this membrane receptor for this particular protein, and it's about how this receptor basically mediates, so it's in the middle of the cellular uptake of vitamin A. So vitamin A referring to retinol. So essentially you have this protein somewhere over here on the outside, and you have this receptor on the membrane, and this thing is trying to go into there and shoot out some vitamin A. So with that in mind, we already have a pretty good idea of what this passage most likely is going to be about. Just by simply taking our time and breaking down the title, we're able to do that. So now we kind of are primed about, we're looking at a protein. The protein is going to this membrane receptor, and the membrane receptor is going to be spitting out vitamin A. So if that's too confusing, don't worry about it. But the strategy is going to be the same no matter what passage you approach. You're going to have to try and prime yourself to understand what these confusing things say. So luckily when we're studying, we have much more time than on the real exam. So when you're studying, definitely take a moment and actually try. It's the only way that you can grow. So once we're done reading the title, the second step that I do is I come all the way to the top and I close my eyes and I try to, again, imagine the file cabinet. And you're trying to pull out terms. It takes about 5 seconds, but it's really helpful when it comes to stamina for each of these sections, because you have, multiple passages, multiple concepts, multiple ideas, sometimes going from one passage to the next without giving your brain some time to flush out all the other noise it's detrimental to your stamina and to your focus. So I just kind of close my eyes for 5 seconds, take a deep breath, and I think about, those few key words like membrane receptors and vitamin A and proteins, and then I open my eyes and I'm now focused. I'm now primed and ready to go to tackle this passage calmly, which is very, very important. Okay. Why is tackling a passage calmly important? Because you want to have as much clarity in your mind as possible, because you certainly don't want to finish this test and think to yourself, I knew some of those terms, but I just forgot it because I was too stressed out. You definitely don't want that to be the reason why you don't score well. So take 5 seconds, read the title, take a deep breath, focus yourself on the key words of the passage, and then jump into it. It's something that I do every single time I take a passage for every section to this day. So let's jump into the first paragraph now. Now, when I'm taking a scientific passage, so what I do is that I read only one paragraph at a time. So I'll read the first paragraph, and then I'll come down to the bottom and I'll try and answer the first question. I won't read the whole passage because in my experience and experience of my students, we've kind of found out that, sometimes you don't even have to read the whole passage. Like I've gone times where I've read only the first few paragraphs and I'm able to answer all the questions. And so we're trying to save some time here by going one paragraph at a time, going to the first question, just seeing how many questions you can answer with just the information that you know already. That's a very efficient way of going about doing it, and this is my approach. So what we do is we read the title, step 1. Step 2 is we took a deep breath and focused ourselves on the topics at hand. Step 3 is to read just the first paragraph. So let's do that. Let's take about 30 seconds or so and try to read it quietly to yourself. That way you can have maximum impact. And then I'll read it with you. So try to read it yourselves, just this first paragraph.
Oh my.
Okay. So if you're a little confused, don't worry. You won't be in a matter of a few minutes. But this is, again, directly from the AAMC. It's a section bank passage 4 from Biological Sciences. So this is saying the majority of dietary vitamin A is stored in the liver and is then transported to a bunch of different organs as retinol by its carrier protein, RBP. Okay, so you have this protein, again, and it carries retinol from the liver to other places. Great. In the liver, retinol forms a complex with RBP known as holo-RBP. STRA6 is the membrane. Okay, so I'm already confused at this point. At this point, I've hit a wall personally to where I can no longer keep all this information that they're giving me in my brain. I have to write something out. So for me, in this case, I would use a diagram. And the reason why I would use a diagram is because it's going to be telling us sort of like a pathway. So you see, in the liver, this forms that with that. This is the membrane receptor for that. And then this binds to that. Once this is done, then this happens. Like it's going to be confusing to kind of hold it in my head. So I'm going to draw a quick diagram. And so we're going to start with the liver, right? So you have the liver, right? And that looks more like a shoe. But you have the liver, and it says that in the liver, retinol forms a complex with RBP known as holo-RBP. So let's say this is retinol, and then this circle is going to be RBP. So the combination of the dot and the circle together is going to be known as holo-RBP. So once we diagram that out, we'll now have a much clearer picture. So again, the dot equals retinol. The circle equals retinol binding protein. And the dot plus the circle equals holo-RBP. Okay. STRA6 is the membrane receptor for RBP. So again, it says that that's the membrane receptor. STRA6 mediated retinol uptake is specific to RBP. Okay, so basically there's some target organ over here. You have a membrane, just like we drew when we were first looking at the title. So we know that this is what the passage was going to be about, basically, was some protein carries retinol through STRA6 or some membrane receptor, and that's it. So it's pretty much the same, which is a good thing. So this is going to be STRA6, so S6, right? And so these guys, holo-RBP, come from the liver and they go to the target organ like so. And this pathway, they're saying this pathway is specific. It is specific to RBP, meaning that if it's not RBP, it's not going to happen, right? If there's no RBP, this thing is not going to happen. So, okay, not going to happen. And uses a transport mechanism distinct from known transport mechanisms. Again, if it's distinct, that means that if it's not RBP, it's not going to happen. So you need the combination of STRA6 and this RBP for this pathway, this STRA6 mediated retinol uptake to happen. You need RBP. So anything else, it's not going to happen, basically is what this says. When it says specific and distinct, that's basically what it's saying. Holo-RBP binds to STRA6 transfected cells Now, what does transfected mean? It just means basically that it has it. So if this cell was not transfected with straw 6, then there's no straw 6 on the cell. If it's transfected with straw 6, then it has straw 6 on the cell. So this cell in our example is transfected with straw 6. And holo RBP, remember the circle and the dot, binds to straw 6 transfected cells with high affinity. What does high affinity mean? It's going to bind very well. There's a large attraction between that occurring. So if you have low affinity, low attraction, high affinity, high attraction, basically. So yeah, again, so it basically means that, you know, it really wants to happen. Only one retinol is provided to its receptor at a time. So when holo RBP is over here, right, it's just going to be spitting out one retinol at a time. So pop, 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 right? So, and RBP remains outside the cell. So, you know, this thing is basically like once this dot, the retinol has come into the cell, the circle remains back outside the cell. So that's cool. Endocytosis inhibitors do not prevent STRA6 mediated retinol uptake. So again, if you try to inhibit endocytosis, right? Basically endocytosis meaning that things from going into the cell, right? If you try to prevent things from going inside the cell, it's not going to work to prevent this process. STRA6 mediated retinol uptake, it's so specific and it's such a high affinity that apparently these endocytosis inhibitors also fail to prevent them. So already we know that if it's not RBP, it's not going to happen. It's very specific, very high affinity, and even endocytosis inhibitors don't work to prevent this thing from happening. So upon uptake from holo RBP, so when these little retinol guys are in the cell, then what happens is retinol is converted into retinal esters by LRAT or stored in the cell by cellular, so CRBP. So either these guys go this pathway or they go this pathway, right? And this pathway is LRAT and this pathway is CRBP. Okay? And that's it. So we've gotten through this whole paragraph. This paragraph is very confusing at first. But essentially, what we did was we took this entire paragraph and we boiled it down into this nice looking diagram, which makes sense to us, hopefully. If you have any questions, please let me know. Any questions you guys have? So far, no? Okay, so the idea again is that retinol forms in the liver. RBP and retinol form a complex known as holo RBP. Holo RBP then leaves the liver and transports the retinol to a target organ, right? This is a target organ, which is transfected with STRA6. And STRA6 is highly, like this pathway is very specific to RBP and STRA6. And no endocytosis inhibitor can block it. And once, you know, the HRBP spits out the retinol into the target organ, RBP floats behind, as we can see here. And once the retinol is in the cell, it either goes to LRAT or it goes CRBP to become retinal esters or to be stored. Great. So now that we've gotten that first paragraph down, we come down to the first question. We don't keep reading the passage. So we go down to the first question, and we just basically see that based upon the question stem, can we answer this question with what we know? So let's read the question. It says retinol uptake was measured in untransfected and STRA6 transfected cells. Okay, so already, what do we know? We know that if it's untransfected, it ain't going to happen. It needs to have STRA6 for retinol uptake to occur. Remember, because it says that It's STRA6 mediated retinol uptake is specific to RBP. So this whole pathway, right, got to have STRA6 and RBP. So if it doesn't have STRA6, well, we know it's not going to happen as compared to cells that do have STRA6. Okay? So we know that. Now it says in the presence of retinol bound to either RBP, okay, good, right? This is good. Or bovine serum albumin. So again, I don't care if you know what bovine serum albumin is or not. We already know that if it's not RBP, it's not going to happen. So, yeah, so this is not going to happen, and this is not going to happen. But transfected and RBP will happen. Uptake of retinol bound to RBP was also measured in cells that were co-transfected with a small inhibitor RNA targeting STRA6. Again, we know that endocytosis inhibitors don't prevent this stuff from working, so it's still going to happen with or without the inhibitor. Maybe just a little bit less, but it's still going to happen. So from this question, guys, and from the knowledge that we have of the first paragraph, can we answer it? Yes, we can. So the question is saying, which graphic shows the expected result of this experiment? So if you guys saw the first webinar, the first recording about cars, you'll know that how I answer questions is as follows. We provide ourselves with an answer choice E. And the reason why we do that is because we need to synthesize our understanding of all this information and boil it down into a few key points. These few key points will allow us to determine the strength or the weakness of each answer choice. Otherwise, we have nothing to compare it to, and we're flying blind and hoping that we remember what we were supposed to remember. So you don't have to write down an answer choice E. I like to do it in my head. But again, what would our answer choice E be? It would be, again, exactly what we just broke down. If it's untransfected, not going to happen. If it's transfected, it will happen only with RBP. If it's bovine serum albumin, it's not going to happen. If it's co-transfected with an inhibitor, it's still going to happen. That's our answer choice E. That is the answer that we would give if we didn't have all four answer choices. If there was only one answer to give, we would give this answer. So now that we are armed with this, the second step to the approach of how I answer questions is to then, again, simply take what we know and compare it to each of the answer choices and just see, is it strong, is it weak? In this case, it's going to be pretty straightforward because it's not as much critical thinking involved. So there won't really be, it'll be very clear cut. So let's just go through answer choice A and look at it. So on the y-axis, we have retinol uptake activity. And then so on the bottom here, we have a negative sign, and that refers to whether or not the inhibitor is there. So if the negative sign, that means that there's no inhibitor. If it's a positive sign, that means that there is an inhibitor. But as we know, it doesn't really matter if there's an inhibitor or not. We're still going to get the retinol uptake, right? Okay, and then the one above it talks about RBP or BSA, and basically this is the binding protein. So, oops, I'm hearing some echo. Whoever just joined, could you please mute yourself? Well, let me just see if I can. Yeah, okay. Seems to work okay now. All right. So, yeah, so again, we're looking at answer choice A, and we have RBP present. We have no inhibitor present, so this should be pretty good. And then the last key bit of information is over here. We have the black bar is untransfected, and the gray bar is transfected. So let's go through each of these categories and look at it. So over here we have the untransfected bar is very low for uptake, and that makes sense. Over here we have the transfected bar with RBP without an inhibitor, and it's very high. That makes sense, right? Using the notes that we have over here. Now, let's move on to the next column. We have BSA, which means that there should be no uptake, period. So this makes sense, but this does not make sense. There should be no uptake. Does that make sense, guys? So we can already cross off A. Why? Because we know that BSA, even if it's transfected with straw 6, the retinol uptake through straw 6 is specific to RBP. So if it's BSA, this is incorrect. Let's move on to B. The first column is the same as A, so we know it's correct. The second column is also pretty much the same as A, so we know it's incorrect because BSA has an even higher uptake, which is even more incorrect. Let's look at C. Again, we know it's incorrect because untransfected should not be there, right? This thing is not right. The black bar should be like it is over here. So we know the answer. We know the correct answer is D, but let's confirm. So this again is the same as A and B. We know it's correct. This is finally accurate because BSA should not be there. And then this one is also accurate because untransfected is low, and this uptake is still present, even though it's a little bit lower because we have the inhibitor. So it's a little bit lower, which I suppose makes intuitive sense. Now, of the four answer choices, D is the strongest, so D is the correct answer here. But yeah, great. So we read only one paragraph, and we were able to break this down systematically and approach each answer choice with the knowledge that we have and the understanding that we have of the first paragraph. Now, instead of going back up and reading the second paragraph, we'll see if we can answer the second question with the knowledge that we have. So keep going. Let's see. Which statement is supported by the data shown in Figure 1? Okay, so we can't answer this, right, because we don't know what Figure 1 shows. But, and here's something that you can take it or leave it. This is something that I do personally. I use priming again. I prime myself again. Why? Because if I read, this is how I prime myself. I read the answer choices before going back up. Why? Again, because that way I will know what the question is asking, so that when I'm reading Figure 1, it will be much more clear what I need to focus on. That's just my strategy. Some students I know don't really like to do it. They like to read the question and go back up to the passage right away. I personally think that you're going to have to read the answer choices anyways. So might as well read it first, get something out of it so that you can arm yourself with some knowledge before you go back up and read the Figure 1. But it's up to you guys what you want to do. So yeah. Let's read the answer choices. Straw 6 activity is associated with this, but not that. Okay? So if this is true in the figure, we will see activity for this, but we won't see activity for that. If we do see activity for both, we know this answer choice is wrong. B says the retinol uptake is only dependent on the ability to bind retinol. Okay, so basically if we have retinol uptake, so only dependent, meaning that if something can bind to retinol, then that's all we need to take retinol up into the cell. But if there's a variance, if there's a difference between the types, like between the amount of uptake, if there's even the slightest difference in the amount of uptake, we know that this is false because that means that it's not only dependent on the ability to bind retinol, it's dependent on other things. So cool. So if basically we see a line, and then we see another line, and another line, and another line, this does not mean that it's only dependent. This means that it's dependent upon other things, because there's this difference, right? So that's B. C says neither this nor that are absolutely required for uptake. Right, so the way this will be shown true is that if we have a line or something on the graph that doesn't have either of these things and still has retinal uptake, then this is true. And D says release of retinol from HRBP is most effective in the STRA6 only condition. Again, if STRA6 only condition has the highest retinal uptake, then this is true. If not, then this is false. So that gives me now I'm so much clearer in what I need to know, but let's go back up now and read the next paragraph. So we just read paragraph 1. Now I'll read paragraph 2, which is basically from here to the caption. And that's enough that we need to understand this second question. We don't need to read anything else. So let's take about 30 seconds and try to read this second paragraph and see if you can remember the answer. Okay. It says that researchers assayed the time-dependent uptake, so time is the independent variable, uptake of radio-labeled retinol. So radio-labeled retinol uptake is the dependent variable. In untransfected cells, so we know if it's untransfected, we ain't going to get much uptake. And then cells transfected with STRA6 alone, STRA6 and LRET, and STRA6 and CRBP. Great. So as we can predict, the control is very low uptake because it's not transfected with STRA6. We see that STRA6 alone is the next lowest, or the next highest. And so if you guys remember, there was an answer choice that said it's the most effective with STRA6 only. So we know that answer choice is wrong. And we know that there is a difference between these different conditions. So we know that the other answer choice that said it's only dependent on ability to bind retinol, so we know that one's wrong. So we can already cross out a couple of answers in our heads. And then it says that the other one was talking about how CRBP was the most effective. Well, we can see that this is the most effective, so that one's wrong. So we've already figured out the answer to the question. And the answer to the question is that neither this nor that are absolutely required, because when STRA6 is by itself, we still get some uptake. So that's the most supported answer. Awesome. Cool, cool. So the caption says, time-dependent uptake of this and that. Note, the activity of STRA6 and LRET cells is defined as 100%. Guys, what that means is that they look at the highest amount of uptake, and it seems to be STRA6 and LRET. So they take this value and they call this 1. And then everything else is compared to that. So this would be, 0.3 or 30% of this. That way the scientists can kind of keep a better track of the data, because otherwise it gets very messy and the numbers get really big. So they're just going to say, okay, we're going to take the highest number, call that 100%, and then everything else will be relative to that. So in case you were wondering what this means, that's what that means. It's called normalization. Okay, but anyways, so we've read the second paragraph, we've read the first figure. We're cruising along, cruising along, cruising along. And again, we already know we can cross off A because it is associated with that. We know we can cross off B because it's not only dependent. We know we can cross off D because it's not most effective in the only condition. So the answer is C. Neither LRAT nor CRBP are absolutely required for uptake, which is true because we have uptake when they're not there. Great. Let's move on to the third question. What is the dependent variable from the experiment shown in Figure 2? Okay, can we answer this question? No. Why? Because we need to read Figure 2. Well, again, let's prime ourselves. Real quick, we're looking for the dependent variable. If it's a chart, where is the dependent variable going to be? Right here on the y-axis. This is the independent variable. So let's go through it. So straw 6 or straw 6 LRAT condition. Conditions are almost never variables. They're conditions. It's like, I mean, nothing's necessarily changing. The conditions will most likely be like, you know, like if you have like a legend on the graph, it's like this is like, you know, this is like, you know, condition number 2, this is condition number 4, or whatever. So it's most likely not going to be A. Retinol fluorescence sounds like a dependent variable. Sounds like something that could change, right? Time is almost always independent because time doesn't really depend on anything for the simplification. So yeah, time is usually independent. And then untransfected cell membranes, again, looks more like a condition. So most likely it's going to be B, but let's go back up to the question, to the Figure 2 and confirm. And we'll go ahead and finish up this passage because we just finished this. So the third paragraph happens to be the end of the passage and happens to include Figure 2. So we will just wrap it up, knock out this passage, and then we'll be done with the questions. So take about 20 seconds, 30 seconds, read through it, just get familiarized, and then I'll go through it. Okay, so as you guys can probably imagine, the answer is retinol fluorescence. That's the dependent variable. So we've already answered that question. But let's read the last paragraph anyways. So the researchers developed another assay or experiment to measure STRA6 mediated retinol release from holo RBP. Okay, so in this assay, different concentration of HRBP were added to membranes expressing STRA6 or a combination of STRA6 and LRAT. Retinol fluorescence, which is enhanced when retinol is bound to RBP. Okay, what does that mean, guys? So retinol fluorescence is higher when retinol is still bound to RBP. So that means that if it's like 100, right? That means that no uptake has occurred. There hasn't been any release of retinol into the cell. So that's why the control, right, which is the untransfected cells, is actually going to be up here, because that means that nothing has been released. So down here means it's been released a lot, because the fluorescence is higher when retinol is still bound to RBP and has not been released. So pay attention to that because that's could confuse some people. So retinol fluorescence, which is enhanced when retinol is bound to RBP, was measured over time. STRA6 and LRAT levels were the same in each reaction. Great. So what are they measuring here? They're measuring retinol fluorescence and the effects of the varying concentrations of holo RBP and they're also measuring the effect that LRAT has on retinol release as compared to STRA6 by itself. So a few things are going on here, so let's break it down. Number one is we have the bigger concentration versus the lower concentration. So it seems as though the bigger concentration does not release as quickly, nearly as quickly as the lower concentration. You see over here, by 3 minutes, the lower concentrations are pretty much, let's say 80% released, right? Whereas over here we have 60% released, and over here we have only about 20% released in the same amount of time. So we know that the less concentration you have, the more it's released. The more concentration that you have, the less it's released in the same amount of time. Which, I mean, I suppose it kind of makes sense. If you think about it, like here's the cell, here's the membrane. Like if you have a little concentration, right? Makes sense. It can kind of handle that. But if you're flooding the membrane with a higher amount of concentration, then it kind of makes sense that it might bottleneck. But either way, this is just what the figure shows us, that the smaller the concentration, the more the release of retinol into the cell. Great. So that's the first thing that we're looking at. The second thing is how much does LRAT help, right? What's the difference between STRA6 by itself and STRA6 with LRAT? Well, when you have a higher concentration, the difference is huge. Look at this gap, guys. The difference is huge. Versus here, the difference there's still a difference, but it's not as huge of a gap when you have a lower concentration. So what can we say? We can say that the higher the concentration of holo-RBP, the more STRA6 depends on LRAT for release of retinol. Fantastic. Okay, so that's pretty much good. It says retinol fluorescence assay in untransfected cells and cells transfected with STRA6, and blah. Note, retinol fluorescence at each time point of each reaction is, here we go, normalized. So remember I was saying normalization, to its own value at 0 minutes, which is defined as 100%. So again, they took this value and they said this is going to be 100%, and everything else is going to be relative to that number. So we got our data, we got our understanding of the passage. So now first of all, we can answer this question. We know it's B, right? Retinol fluorescence. Which statement regarding STRA6 mediated retinol uptake is best supported by the passage? So when it says best supported by the passage, guys, it's not talking about figure 2. It's not talking about figure 1. It's the entire passage. So basically, when I look at this question stem, best supported by the passage, it's basically AAMC's way of telling me, hey, the answer to this question is in the passage. And so it's healthy to remember that. Let's look at it. A says retinol uptake activity by STRA6 is limited without the presence of intracellular binding proteins. So is that a strong answer? Yeah, it's a strong answer. So let me pause real quick and refresh you guys. So again, in my mind, we have certain characteristics that we know are true. We know that STRA6 is specific to RBP. We know that it is stronger when you have LRAT. Then it's stronger when you have CRBP. And we know that the more holo-RBP that you have, the more STRA6 depends on LRAT. So we have all these different characteristics that we know that are true about the process of retinol uptake through STRA6. So what we're doing is we're taking our understanding and we're going to be comparing it to these answer choices to make sure that we see how strong or weak they are. So A says that uptake activity by STRA6 is limited without the presence of these LRAT or CRBP. Is that a strong answer? Yeah, so let's leave it. But here, if you guys didn't see the first recording, the first webinar, this is the next most important strategy that I use to answer questions. Is before I just cross out things when it requires some critical thinking, when it asks questions like which is the best supported, whatever. When it requires this critical thinking, I don't like to cross off answers one at a time. What I like to do is I like to say, okay, based upon, my answer choice here, like based upon my understanding, A seems like a strong answer. Okay, does that mean that I can cross it off? No. Does it mean that I can select it? No. What do we need? We need to know, compared to the other answer choices, which one is the winner. Now, the way I like to think about this is like March Madness. My alma mater, UNC Chapel Hill, won college basketball last tournament. Does that mean that they're the best team ever? No. It just means that of the teams that they were bracketed against in the tournament, bracketing referring to this, right? Of the teams that they were bracketed against, they were the winners. So what we do is we take that bracketing and we apply it to this strategy. So answer choice A, it's a competitor, it's strong, but let's see compared to B. Is A stronger than B? And if A is stronger than B, then we know it will never be B, because A will always be stronger. That's a much more effective and logical way of doing process of elimination that I was never taught. I don't know if I missed a class or something, but it's something that I had to figure out. But it works tremendously. So A versus B. So let's look at B. The expression of straw 6 is likely to be higher in the liver than in target organs. Well, we know that straw 6 is on the target organs, not in the liver, and either way, A is definitely stronger than B. So we can cross off B. Now here's the last tip. The reason why I can say A is stronger than B, because you might be thinking to yourselves, well, how do I know which one's stronger or which one's weaker? I use what I call MSLR. MSLR stands for most support, least rationalization. Most support, meaning this statement has a lot of support in the passage directly. Least rationalization says I'm basically spending less time understanding the answer choice. So with B, I have to I always find myself rationalizing whenever I have to look up and to the left. If you guys think about whenever you're answering questions, if something's very simple and straightforward to you, like answer choice A, you'll be like, oh yeah, okay, that kind of works. But then B comes around and you start to think to yourself, oh my gosh, are they tricking me? Is this a trick question? Maybe I'm going to outsmart the AMC. Maybe they want me to be thinking that it's A, but it's not A, it's actually B. We start to rationalize these things when in reality, that's exactly the opposite of what we need to be doing. We need to find MSLR, most support, least rationalization. So sometimes the simplest answer is the correct answer, even though we want it to be harder, right? But the AMC is not trying to make this very ambiguous. Trust me. They're supposed to be relied upon by all these universities to become the standard that they look at to admit students. So that's their job. The test writers' jobs are to make things clear that can separate the strong candidates from the weak candidates. The strong candidates can see through these sort of tactics, can see through and see the clear, reasonable answer. Those are the candidates that they want to admit into their school. People who can think critically, think for themselves, because when you're gone, when you're out of undergrad, when you're out of even medical school, I mean, you're by yourself. You got to be able to handle problems by yourself. You have to be able to at least know how to solve them, how to approach them, what relevant data matters, what doesn't matter. And so it's a simple thing, but just learning how to answer from this way is a skill that you'll carry through for the rest of your life, whether it's in your relationships or whether it's in your career or whatever it is. This method of problem solving, I mean, it's what makes critical thinkers critical thinkers. So you're able to see the most support and least rationalization. Now we can cross off B because we know that A is stronger than B. So let's look at A versus C. Which one wins? Retinol uptake activity by straw 6 is accelerated when retinol is stored as intracellular retinol rather than as retinol esters. So we know that this is basically saying retinol uptake activity, is it higher with CRBP or is it higher with LRAT? Well, we know that it's higher with LRAT than it is with CRBP. So we know that this is not as strong as A, because this is saying that it's higher with CRBP, not LRAT, which is wrong. Now let's look A versus D. D says binding of holo RBP to straw 6 is likely to have a Hill coefficient greater than 1. So before you guys get scared about what Hill coefficients are, please remember this answer choice is not stronger than A because it doesn't have the most support and it requires a lot of rationalization. So we can cross off D because A is stronger than D. A has the most support and the least rationalization. So don't even freak out whenever you see Hill coefficient. Oh my God, is it me? Like, did I not know something? Did I not study something? Should I have known? No. It's asking you which is best supported by the passage, which means that the answer choice is in the passage explicitly and supported by the passage, and it has the least rationalization. So this one, don't worry, it requires too much rationalization. Yet, however, a lot of students will pick D because they think that somehow they missed something. And all of this stuff, they just basically give up and throw out the window because they are more anxious about not knowing something than they are about sticking to the most support and least rationalization. So A is the correct answer here. Let's keep going. Almost done, guys. I think we have 2 more questions. Yep. From the data shown in Figure 2, great. So again, this question stem is basically the AAMC's way of telling me, okay, Nicholas, the answer to this question is in Figure 2. Where does it, sorry, we got a question. Where does it say that straw 6 is accelerated with LRAT? So over here in the first figure, we have a massive difference in the uptake activity between LRAT and CRBP. Massive difference, almost a 70% difference. So that's how I'm knowing that LRAT is stronger than CRBP, because the Figure 1 determined that. Does that answer your question? So yeah, uptake activity, and then they were showing the different pathways that RBP to straw 6 that read. But in Figure 2, yes. But in Figure 2, yeah, in Figure 2, it's confirming LRAT's efficacy as well, because remember, 20 doesn't mean low uptake. It actually means high uptake, because the dependent variable here is fluorescence, not uptake. And fluorescence is enhanced when retinol is still bound to RBP, meaning that the lower the fluorescence, the higher the uptake. The higher the fluorescence, the lower the uptake because it's still bound. So when we have LRAT here, it's always pushing it towards more uptake, always. Look at the difference here and here. When you have LRAT, it increases the uptake in 3 minutes by about 50%, from 10% uptake to about, 60% uptake. So LRAT in this figure as well is shown to be increasing the uptake of retinol quite significantly. Makes sense? Okay, great. Awesome. Awesome. Yes. Yes, and this is something, guys, that you have to understand that for example, when I'm reading this sentence here, the reason why I'm able to catch this is because when I read things, even if I can't read out loud, I will use hand gestures. And in my mind, I'm going to slow down. I'm going to even use my finger and point on the screen and trace my finger along so I don't miss it. Retinol fluorescence, which is enhanced when retinol is bound to RBP. And then I don't keep reading that, guys. I stop because it's a bit of a confusing statement to make. And so I have to process it. And this is just me. Maybe you guys can read this and understand it faster than I can. But the point is that you got to actually understand it before you keep on reading. Otherwise, you're going to drown yourself in knowledge. You're going to drown yourself in information. And what's going to happen is you're going to forget what this graph is even showing you. And these little phrases, I mean, misunderstanding one little phrase, one little word, can make or break your understanding of the figure or of the whole passage, which is crazy, but it's important that you slow down and kind of understand what is this saying. So where are we at? From the data shown in Figure 2, which statement best explains the? By the way, good question, M. It's a good question. Which statement best explains the relationship between STRAW-6 and holo RBP concentration in retinol release? Okay, guys, this is exactly what we were doing when we were analyzing the passage, or the figure. Remember, we were looking at two things. The Figure 2 showed us two things. Figure 2 showed us the difference between when you have a high concentration versus when you have a low concentration. And it also showed us what happens when you add LRAT. How much does it help? So we concluded that when you have a high concentration, there's less uptake. When you have a low concentration, there's more uptake in the same amount of time. When you add LRAT, with a high concentration, LRAT helps tremendously. When you have a low concentration, it still helps, but not as much. Okay. So that's what we're going to arm ourselves with to understand this question. So that's basically our answer choice E, was those few statements that we just made. So let's look at each answer choice and see the strength of it. So answer choice A says STRAW-6 catalyzed retinol release increases as the molar ratio of HRBP to STRAW-6 increases. So basically this is saying the more concentration you have of this, the more uptake you have. And guys, compared to what we just said, no. The lower concentration of HRBP yields the most uptake. So, nope. Retinol release in the STRAW-6 alone condition is not affected when you increase. Yeah, this is ridiculous. So, guys, again, this is basically saying that when you have STRAW-6 by itself here, you have STRAW-6 by itself here, is there a difference in the activity? Absolutely, a massive one. So this statement is false. This statement is saying that there's no difference, which is crazy. C says STRAW-6 becomes more dependent on LRAT for retinol release when the HRBP to STRAW-6 mole ratio increases. Yes, absolutely. This is absolutely true. Remember? So when you have more HRBP concentration, it's more dependent on LRAT. So we go back up here to look at it again, just to show you. Remember the difference, let me clean this up. This is very, very messy and it's causing me some anxiety. So hold on. Again, when you have a high concentration of HRBP, the difference between STRAW-6 by itself and STRAW-6 with the help of LRAT is huge compared to the difference over here. So again, what the statement is saying is that as you increase the concentration of HRBP, STRAW-6 becomes more dependent on LRAT for uptake, which is true. So let's look at the last answer choice, which is D. And D says retinol release is only dependent on the presence of HRBP. And as we know, it's dependent on more than just HRBP. Otherwise, STRAW-6 uptake would not have changed when you have LRAT versus when you don't have LRAT. So let's see. Last question. Here we go. Looks like an easy one. The organ in which the HRBP complex forms also functions to what? Well, what organ is it forming? It's the liver, right? So this question is saying the liver also functions to what? Does it function to store bile, secrete glucagon, produce HCl, detoxify drugs? Guys, if you can enter that in your chat, or you can unmute yourself and tell me the answer, that'd be much better. So let me know what you guys think it is, and then I'll give you like 20 seconds to figure it out. The liver also functions to store bile, secrete. So Sandra, you say D. That is correct. Great job. Fantastic. Now, Sandra, I'm going to put you on the spot. No, I'm just kidding. But basically, guys, what is B? B is the gallbladder. Yep. Secrete glucagon is what? Pancreas. HCl is produced where? No clue. Pretty sure it's the stomach. But fantastic. So there you go. So this is gallbladder, this is pancreas, this is stomach, this is the liver. Great. Okay. That's it, guys. We got 100% on this passage, and yeah, this is a passage that I intentionally choose because it looks so freaking scary. Like when you look at the figures and everything, you're like, what is this? Like, what are we supposed to be knowing here? But hopefully by kind of breaking it down with me, it's a little less scary, which is the goal. Because the less you're afraid of something, the more courageous you are to go ahead and tackle it. Because you can figure this out. I mean, look at this. Like, we figured all of this out, and it doesn't require such a vast amount of content knowledge. What it requires is an understanding of the passage. So let's clean up this thing here. Any questions that you guys have while I clean this up? Any questions? Anything stuck out? Anything that wasn't really that clear? And let me unmute people. I know, Sandra, you're locally muted. M, you are not muted. I don't know. It doesn't look like you are. But, yeah, if you want to say hi, ask questions like this, it could be easier than typing it. But yeah, okay. Oops. There we go. So while I clean this up, any questions that you guys have, just type away or ask me on the phone. And I'd be glad to answer them. Doesn't seem like there's many questions. It seems as though, oh. On the actual test, how can you manage your time to do all this? The question was, or does it just come with practice? Absolutely. Speed comes with practice. So you have two options here, okay? You have option number one. Let's zoom out for a second. On the actual test, you're going to be prepared, okay? You're not going to take the actual test until you're prepared. So the real question becomes, how do you prepare? When you prepare, is speed more important, or is strategy and consistency more important? In my opinion, strategy is more important than speed. Why? Think about any skill that you guys have. Think about anything. I mean, riding a bike, okay? When you first hopped on the bike, like, was it like 2 seconds, like hop on, boom, and you're gone? No, it wasn't. It was training wheels, first of all. Somebody probably lifted you up and put you on the bike, and then you like got the courage, like, okay, here we go, pedal, 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 pedal. That's why speed is not as important. Because when you're developing a habit, when you're developing a skill, when you have this clock or this timer on you, you're rushing yourself, and you're going to go back to the bad habits. And when I say bad habits, I mean habits that are counterproductive to your goals. Habits that do not lead you to an understanding of the strategy that will help you ace this test and to just, in general, become a better problem solver. So how are you managing the time? Exactly what I just did. If it takes you a half an hour to get through a passage, as long as you're practicing consistently and you're analyzing consistently and you're studying and drilling what you didn't know from this passage consistently, over time your speed is going to get better and better and better. Now, give me 1 second and I'm going to explain that in a different way because it's a very, very important question. You know, a lot of times students will when I tutor, they come to me and they say, hey, I just finished a full-length exam. I'm like, okay, how'd you do? They're like, well, I'm like, did you answer every question? They're like, yeah, I answered everything, and I didn't do very well. I'm like, in my mind, I don't like that. I don't like it that people finish the exam. I mean, it seems as though they're finishing the exam because they don't want to waste it. when in reality, by forcing yourself to finish the exam, you are wasting it. Because you're not practicing the strategies that you're learning consistently enough. So, in my opinion, when you're first starting out studying, don't worry about finishing an exam. Like, you don't even have to take an exam. Just take a bunch of passages like we're doing right now, go to the AAMC section bank, and start practicing with these passages one at a time. Then when you get comfortable with the strategy, then try your best at an exam, but it just depends upon how much time you have to study. Some people rush themselves and they take only 4 to 6 weeks to study. Others, they give themselves more time and they can learn more and build stronger habits. It just depends upon you and the amount of patience that you have for doing this. So speed will come with practice, and it's better to understand the strategy before you understand how fast you're doing something. So when I say that, now that I'm done cleaning up this board here, let's go quickly back to this presentation. So when we go to the boogeyman method, what we just completed, guys, was practice. That's what we just did. We just spent some time and we did our very, very best to get through this passage. Step 2 is to analyze the passage. So we're going to go through and analyze the passage, and we're going to ask ourselves a few questions to make sure that we did the strategy well enough. And if we didn't do the strategy well enough, well, we've learned, and we'll make a mental note of it, and we'll be better for next time. And another part of the analysis is to circle the terms or phrases that you didn't understand very well. And for example, you can ask yourself, which of these terms or phrases, in hindsight, if I knew these terms or phrases in hindsight, would this have helped me get this question right? Would this have helped me understand the passage a bit better? And we're just going to go through really quickly and circle the different terms or write down the different terms in a spreadsheet. And then from there, that's basically formulating our flashcards. It's like automatic. So we practiced, and then we're going to analyze and break down our practice. We're going to organize those terms and phrases into a spreadsheet, and then turn those terms or phrases from the spreadsheet into flashcards, and then we're going to drill those flashcards. And then once we're done drilling, we're going to go back to practice. So Sandra, to answer your question again, when it comes to how do we develop the speed, it's going to come by following this process diligently. So at the beginning, I know for me, at the beginning, it might take you a week to get through, a couple passages, analyzing those couple passages, organizing those couple passages, and then drilling. But you're going to get faster and faster at this process to the point where, you can get a full-length test done in a day. And then that night, you can start to analyze it. The next day, you finish analyzing it and start to organize it. And then the next day, you drill the flashcards. So like in 3 days, you can get through a full-length test, the analysis portion, the organization portion, and drilling the concepts that you learned from that full-length test in a matter of a few days. So like, it's not going to happen overnight, but by diligently following the process or the strategies or whatever, yes, you will absolutely get faster and faster and faster. And it's very freeing to know that. So now that we're done practicing, we're going to go ahead and analyze the passage. So from here, just a brief, we don't have to spend so much time on this, but just briefly kind of picking apart the passage. So majority of dietary vitamin A, so stored in the liver, transported cellular target organs as retinol. So if you didn't know what retinol is, it's vitamin A kind of now. But you can always add that term, what is retinol? It's up to you. Retinol binding protein, same thing. But we pretty much understand what these things are from the passage. So just keep kind of perusing through this passage. Holo might be a prefix that is confusing. I don't really know what that means, so maybe that's something to look up. STRA6, membrane receptor. So if you didn't know what a membrane receptor was, then you could study that. Uptake specific. So this word specific, that seemed to be important, guys. So specific, when you make a note of that, you can say like, what does it mean when something's specific? And it means that this mechanism is only going to happen with, for example, RBP. Distinct. So transfected, that was an important term to know. If you don't know it, now you should. High affinity, KD wasn't really that important to the passage, but it might have helped you understand a little bit more. So you can do that. If you don't know what nanometers are, nanomolar, you can do that. Endocytosis inhibitors, you can ask yourself, what are those? Retinal esters, you can ask yourself what that is. What the heck is an acyl transferase? You can look up that. Radiolabeled retinol, what does it mean to radiolabel something? What else? What else do we not know? No. Normalization. I think normalization is an important one because you're going to see that time and time again. So look at that. Like we got normalization, we got, radiolabeling, we got acyl transferases, retinal esters, we got endocytosis inhibitors, we got high affinity, membrane receptors, holo, transfection, we got specificity, we got, retinol, we got plenty of different terms that we can now organize. And the way I do it is and we can even go down into the questions, which we should be doing. So we keep going through these, like anything from here, like over here, like you can put, when you have plus or minus in a graph, what does that mean? It means that minus means you don't have it, plus means you do have it. What else can we learn here? BSA, right? What the heck is BSA? And everything else is pretty much, oh yes, Hill coefficient, right? Greater than 1, so these different things. Oh, and then over here, like 4 different things here. So we got a pretty good set of 15 to 20 different questions that we can know just from this one passage. And so if you're talking about trying to get the most bang for your buck, I mean, this is the way that I analyze passages. This is the way that I study. Is I wring the potential out of these passages dry. I wring them dry. And that's how I was able to kind of progress so rapidly, is because I was just, honestly, guys, it has to do with honesty. It has to do with honesty. Did you do your best in the passage, yes or no? Yes? Awesome. That means that your failures are failures that you can learn from. Okay, what were all your failures? Select all of them. What did you fail to understand? Awesome, okay. So now, once we're done with this is the analysis portion. We just finished that. Actually, sorry. So that's part of the analysis portion. The other part is right here, the checklist. And this checklist, guys, is basically what I ask myself just to kind of make sure that I'm studying this strategy. And the checklist is did I read with emotion? What I mean by that is, whenever I'm reading again, I'm trying to slow down, use my finger to point, enunciating words, emphasizing words, trying to read not so monotonously, because when you read with emotion, things stand out to you more. You start to care more about the passage. You start to see things that are more clear. So if I did, awesome. If not, don't worry about it. We're going to skip number 2 because that's only for cars. Number 3, did I pay attention to the question stem? Again, very, very important because, for example, in question 25 down there, it says in figure 1, sometimes we might get confused and think that the answer choice is just in the passage somewhere, when in reality, the correct answer is going to be in figure 1. So that's what I mean when I say question stem. Same thing with, this one, figure 2, best supported by the passage. That's a question stem. So if I missed those question stems, if I didn't make a conscious note of it, then I know that next time I take a practice passage, that's something I need to focus on. Number 4 says, did I use bracketing to eliminate answer choices? A lot of these answer choices were very straightforward, but for example, we did use bracketing to answer one of the questions, which is great. If we don't, if we find that we're not doing this, then again, that's another strategy that you can work on. The 5th one over here, when determining the strongest answer of the bracket, did I use MSLR? So that's very straightforward. Did you do it or not? Did you get tripped up? Did you start to second guess yourself? What happened? All these different minutiae, all these different details make or break the difference between you understanding your mistakes and you having to, be frustrated about not knowing what's going on. Like, you put in all this work and why are you still not succeeding? Well, these little minutiae could be the questions that you could ask yourself to find out whether or not you're messing up with the strategy. And the last one is, did you come up with an answer choice E? Very, very important. That's basically, answer choice E is basically your understanding of what you just read. So if you didn't come up with answer choice E, that just screams sort of either you forgot to do it or you had a lack of confidence in your understanding of the passage. So if that's the case, again, you have more direction on what you need to focus on for next time. So now we're done with the analysis. The analysis is basically 2 parts. 1 is highlighting the different things that you didn't understand, and 2, this is about the content. So analysis has 2 parts. 1 is kind of highlighting the different content that you didn't understand, and the second is kind of understanding the different parts of the strategy that you failed to implement. So once the analysis is done, okay, now we put them into this bucket. And this is a bucket that I just kind of, it's on my website. You can use it for free. It's all under resources. If you see, we have flashcards. The MCAT spreadsheet is what you're just looking at. I have a PowerPoint for that, and yeah. So if you just come here and you click on spreadsheet, this thing will kind of pop up, and you can copy and paste it. But essentially, this is how you're gonna organize yourself. So this is the 3rd step, which is to organize. So you organize all the content. Basically, you go through, and today is July 30, and then problems, test location. This is under biological sciences. This is the section bank passage 4. And then what are some of the questions that we can add to Quizlet? So first question that comes to mind is, what organ secretes glucagon? Yeah, hello? Oop, I think somebody needs to be muted. There we go. So yeah, what organ secretes glucagon? What organ stores bile? What does it mean to have a Hill coefficient greater than 1? Etc., etc., etc. What does transfected mean? What does it mean when something specific? What does an endocytosis inhibitor do? So you basically come up with the questions and you organize them on this spreadsheet. So that's that. So that's basically this organization step. And then from there, you take what's on here, you close up your passage, you open up Quizlet, and you kind of, what I do is I like to use Quizlet because of this function called learn. And learn is an incredibly powerful tool. Basically, what happens is that when you make a flashcard, for example, we'll come in here and we'll make the flashcards. So this one will be, again, the term is what organ secretes glucagon, and then we'll say pancreas, and then over here we'll say what organ, what else did we say? What organ stores bile? Stores bile. We'll say gallbladder. Okay. So then what you do is you go ahead and you make your flashcards, and it's basically exactly each question. The reason why I prefer to do one question and at a time is because like I used to make the mistake of putting like, what's this and what about that and what about this and what about that onto one flashcard, and it got way too confusing. So one question per flashcard, keep it simple, your brain can understand it. This is for you to help, it's not to make you more confused. So keep it simple, break it down like that. What organ secretes glucagon? Pancreas. Boom, easy answer. So then what you do is, that's the third step, which is organize. So we've practiced the passage. We've broken it down for the content we can learn, we can extract from the passage, as well as the strategy checklist to make sure that we're doing the right strategy. And we're organizing those content in the form of, this spreadsheet, taking that, making them into flashcards. The fourth and final step of the method is to drill what you just created. And the way I like to do that personally is by using this learn function. So you come in here and you hit learn. And then, I put pancreas. That's funny. So what you want is you want to go to options. And you want to select to prompt you with the term. I don't know why it starts you with the definition. It's ridiculous. But start the thing, prompt yourself with the term, hit X. Then you'll have a question, and then you'll type the answer, right? You'll hit answer, it'll be incorrect. You can override if you were right, and that will put it in the correct bucket. Or you can edit this, if you feel like you need to edit it. So, what organ stores bile? Okay, gall, bladder, right? Hit answer. Correct. So that goes into the correct pile. Press any key to continue. What organ secretes glucagon? I know I wrote pancreas, so I just want to get the answer right. I'm going to edit it now. I'm going to put pancreas. Save. Great. Okay, then this one is again something blank. Press any key to continue. Done. So look, now it shows us round 1 is done. Round 1 we have correct 50%, incorrect 50%. Press any key to continue. Okay, so look what it does. It starts me off with the incorrect stuff. It doesn't give me the correct stuff anymore. So we basically put down this one, answer, correct. One more question. This one, answer, correct. Round 1, we got 50%. Round 2, 100%. So now we can star these terms and start over. Or we can start the whole thing over. But for me, I'm done. That's it. So for you guys, you take as many rounds as you can. And for you, it might be like, a couple hundred flashcards, maybe even like 500, 600. But you'll go through the round, and the first round might be like 65%. So you're not going to do those correct ones again. Automatically, it will give you the incorrect ones, and you'll go through, and you'll take as many rounds as it takes to answer these questions. Now you guys might be thinking, okay, so that's basically drilling, right? I mean, it's a very simple flashcards, but for some reason, for me, it took me a while to figure out the power behind this, and that's what I do. So I take as many rounds as I need to get through all the flashcards correct once. Once I have successfully completed to get to the last round is 100%, once I've successfully completed that, the whole deck, I go back and I practice more passages. Maybe I'll take a full-length test if I'm ready. And then I just keep going from there. So this process is scalable, meaning that it works for a small passage for a few questions, and it can work for an entire full-length test for many, many, many hundreds of questions. And the process is the same, and that's the most effective way that I found to do it. So yeah, so now we've gone through 1, 2, 3, 4, and 5. Number 6 right now, we've got about 20 minutes left or so. Any questions on the entire process of studying? Anything that's not clear? Again, there's some resources here. We can have flashcards right here on my website in case you want to get a head start. I've embedded a bunch of flashcards on here. So like if you want to study like behavioral sciences or something, you can just click on the behavioral sciences button, and it will, once it loads, it will bring us down to that section of the page. But any questions about the process? What book or what material was used for those flashcards? So that's a good question. Oh, there it goes. Yeah, so it brings you down to that. So, for example, you know, my favorite resources are free. I believe that you shouldn't spend money on a book if it doesn't give you questions or practice passages or full-length tests. The reason being that the internet pretty much solves a lot of this problem. Now, the resources that I use are, one second, I'm going to pull up a list, and I can email this to you as well. But basically, where is it at? This one. I use, you know, Google is the first option I go to. Like, it's very straightforward. You know, you have what organ secretes glucagon. So instead of opening up a book, I literally just type it into Google, and in about 30 seconds I get the answer. So Google is the first place I go to. Second place I go to is, you know, the articles that Google gives me, right? Like the front page, the first page of the results. That's another resource. Another resource that I really like is aklectures.com. I think aklectures.com, the dude spent so much time explaining a lot of these concepts, so, in my opinion, at such a great pace, very slow, very methodical, very comprehensive, and it's all free, that I think that it's actually better than Khan Academy. But that being said, AK Lectures helps, Khan Academy helps. I suppose it's not going to be working here. Oh well. So yeah, I will definitely send you the list of resources that I use. But basically, it's those. It's AK Lectures. It's Khan Academy. It's all these different things. But first and foremost is Google. Just Google it. It's a good question. Any other questions? Dave, Sandra, Bianca, any other questions? So this means that either, oh, here we go. All good, thank you. Okay. All good, yeah. So these things are going to take a bit of time to learn and understand. But essentially, the more you kind of dive into it, the quicker and quicker and quicker you get at this, the more, you know, you understand the strategies, the sharper you become at the strategies. The more you drill and organize and analyze the passages, the more you get stronger and stronger at the concepts. And it's just, yeah. You started with process to study. What is your process to study, or any advice? So seriously, yeah, you're right. The number one thing is a process to study and a motivation to commit. So the process that I used to study is literally what I just showed you guys. It's exactly this. And I'll break it down to what it looked like for me, you know, because like, so I started this way of studying for about 10 weeks. 10 weeks and I gave myself about 500 hours. So what I did was I bought the resources that I could. I bought Next Step 10-pack test bundle because it was about $250, and it gave me 10 tests, and you can take each test 5 times. So that's like upwards of 50 times you can practice a full-length test, which is enough. I also bought the AAMC full-length test 1, and I bought the sample test. The full-length test number 2 had not come out for me. It is out for you guys, though. And I purchased the section bank, and I purchased the question packs. So literally every AAMC material I could get my hands on, I bought. Why? Because, I mean, they're the guys who write the tests, so I'm sure they would be able to give me the most accurate practice. So that's what I did. I bought those bunch of those tests. I started with, you know, for me, cars was a really, really, really low point. Like, you know, I think it's on here as well, but basically I was like really happy about my cars. So you can see I started it back in 2014 when this was like the different scale. I got a 52nd percentile for cars, and I improved it. My second time I scored it a couple of years later, I got a 98th percentile. So I was really proud of that one. And you know, it was just about basically this checklist that I used. You know, I used a lot of logic and reasoning to cut out through all the noise that everybody was telling me, all the advice that didn't really make sense to me. I was always questioning everything, and these different strategies kind of paid off. So what I would do is, because cars was my weakness, I started my day at 8 a.m. Because I had just quit my job. I quit my job because I had enough money in my savings to really take the time. So I quit my job. I spent 10 weeks, and I would study the same time that I would take, like people take the actual test, which is 8 a.m. So that was my logic, was to start studying so I can train my brain to like be in that mode of thinking by 8 a.m. every morning, so that after doing this for 10 weeks straight, by the time I took the actual exam, it was just like another day at the office. So I would study. I would like wake up around 6:45. I would fill up this is in the summertime in Arizona, so it's very hot. So I'd fill up a nice big bottle of water, and I would head out to the park. I would walk to the park, do some pull-ups, do some push-ups, do some sit-ups, get my blood flowing. Then I would sit underneath the shady tree and just meditate for a little bit, you know, think about other things than the MCAT, or maybe I'd think about the MCAT, I don't know. I would just let my thoughts wander, and I would enjoy the nice greenery, the nice shade, and sip my water, and then say hello to the people passing by in the park. Most of them were old ladies walking their dogs, so, you know, they didn't really remember me, even though I was there every morning. But yeah, so I would do that, and I would walk back home. I would stretch, do a little bit of yoga, and then I would jump in the shower, freshen up. I wouldn't eat anything because I just don't enjoy eating in the mornings. For example, I still haven't eaten all day today. It's just something that works for me. I like to eat breakfast in the afternoon. So that's what I would do. So then, you know, I would shower up, and then I would sit down at my desk around 8 o'clock. You know, I wasn't super strict about it, but I tried to do it. And then what I would do is I would start with a cars passage. For me, it was what I need to work on the most. So I just like, all right, my brain is refreshed. I have the most focus. I have the most energy. I have the most willpower. I'm going to do the hardest thing for me. And the hardest thing for me was, at that time, cars. When it became easier, then I switched my focus towards biological sciences, and then behavioral sciences, and physical sciences. In my opinion, I didn't give it the respect it deserved. I have a degree in chemistry and all that stuff. I actually like those concepts, but I focused more on my weaknesses, and that's just ended up what happening was physical sciences ended up becoming my lowest score, which is ironic. But anyways, so yeah, you know, I would start off with the cars passage. I would warm up with one. I would not do it timed. I would do it again according to the strategy, according to the method that I had established. And, you know, once that's done, then, you know, I'd be like, okay, now it's time for me to do a few passages. So I'll do a couple passages as much as I had time for. For me, because I had more time, I studied 8 hours a day. What I did was I would do practice. Like I would do this first step, practice, and I would practice literally. It wasn't a set number or a set number of sections. It was whatever sections I felt were giving me a hard time, I would do as much practice as I could. So it might be like, you know, from like, you know, 8:30 or 9 o'clock, I would study until 3:30. with, some breaks in the middle, a lunch in the middle, essentially the same schedule that the actual MCAT test is. You have 8 a.m. to, 9:30, then you have a 10-minute break, 9:45 to this, then you have a 30-minute lunch break. That's how I studied pretty much, and it was like from 8 a.m. to 3 o'clock-ish. And so I would do as much practice as I could. I would practice everything. I would do, physical sciences. I would do biological sciences. And the passages that I pulled from were what I'm showing you guys right now, the section bank and the question packs. So I would do that until the time was done. Sometimes I got in more passages, sometimes I got less. But I just practiced, and that's it. And then once it was 3 o'clock, I stopped practicing. I took a nap. I woke up. I worked out. I cooked dinner, ate dinner, watched some TV. And then around 8:30, it's like 5 hours later, I jumped back on my desk and I started analyzing. So I'd break down the passages that I worked on earlier that day, and I did exactly what I showed you guys. So I would pull out, the terms, and I would put them in the spreadsheet. And I would also go through the checklist to make sure that my strategy was not weak. Did I forget something? Almost always I forgot something, without failure. So I would go through and I would analyze for strategy, and I would analyze for content. And then step 3 is to organize it. So I'd organize all that content in here, turn them into flashcards, and then usually the night was done before I finished organizing. So I would continue it the next morning. So like from 8 a.m. or whatever, I would continue. Like after I warmed up with the cars passage, instead of practicing more, I would finish up the cycle, which is something that people find is different. So I would not continue practicing, except for cars, until I finished analyzing and organizing. So once I finished that, the next day, maybe it took me a couple hours the next day, then I would start drilling. And again, I would not move on to practicing if I do not finish drilling everything, as many rounds as it takes, till I get 100%. Because trust me, guys, when you start doing this, if you don't complete the cycle before you continue to practice, you're going to drown yourselves in the amount of content. I mean, you're going to get 400, 500, 600 flashcards made, but you haven't drilled it, and you're going to get less motivated to drill it because it's a very daunting task to have to go through flashcards for the first time ever in your life and have to go through 600 flashcards. So I fortunately was diligent enough, and I actually enjoyed it. It actually freed up a lot of concern about, like, what do I do here? What do I do here? No, I just follow the cycle, follow it, and I can spend less time worrying about how to study and more time actually studying instead. And so, yeah, so I would practice, from 8 a.m. to 3 o'clock. I would analyze and organize whatever I did that night, and it could bleed into the next day. And then, once that was done, I would start drilling it, and I wouldn't stop drilling until I got through as many rounds as it took to get through that last 100% figure and make sure that everything I've answered correctly at least once. And then I would go back to the top and start practicing more. Once I got good with the strategy, I said, okay, now it's time to take a full-length test. And so this maybe was after a couple weeks, then I started taking full-length tests. So like if I have 10 weeks, after week 2 was completed for me, was when I felt comfortable with the process, with the strategy, to where I was like, okay, I'm going to go ahead and jump on that next step, test prep 10-pack bundle, and start churning out full-length tests. And then again, following the strategy, following the process. And I think by the end of it, I think I took I took quite a few practice tests, guys. It was quite a few. But, that's basically the process. And so, so for the remaining 8 weeks out of the 10, I was doing full-length tests, analyzing those full-length tests, which took days to do, organizing those full-length tests, drilling those full-length tests. And remember, the flashcard decks that you make, they're cumulative, so they get more and more and more and more. But you don't need to. Like if you find that you know a concept pretty well, just go to Quizlet, go to your flashcards, and delete them. Just take this and delete the flashcards. So yeah. So you can edit this. Where is it at? Add or remove terms, right? You can remove the terms if you feel like, okay, this is kind of boring. I think I get this. So just go ahead and, you can delete. delete this one. We can delete this one, and we're good to go. So you can always add images here. You can add different things. You can hit auto define, which says no results found. But anyways, that's what I did, right? So like the flashcards are one deck; they're cumulative. So essentially, that's the method that I did up until the test, and then by that time I was used to getting up at 6:45. I was used to studying at 8 a.m. I was used to taking full-length practice exams. I had studied a lot of these concepts. I was really familiar with the strategy. It was habitual at that point, and that's the name of the game. And not only that, but like an important thing is that, again, I had the motivation to commit. I knew exactly what I was doing it for. I was doing it to get freedom. I had enjoyed the process. Again, I was getting up, I was doing yoga, I was like going to the park, working out, I was cooking dinner, napping, I was watching TV. This is my story, this is my process. I took 10 weeks. For you guys, it might take 15, 20 weeks. It might take 4 weeks, 5 weeks. I mean, it's completely subjective. But it's important to understand these two things, having a process to study and committing to it, finding the motivation to commit to it. And just being honest with yourself along the way. If you feel like you're not ready to take the test, don't take the test. Get your refund. Or if you can't get your refund, then take the test and void the scores so you get more practice. I mean, there's no rule, there's no law that says you need to take the test after 10 weeks of studying or 12 weeks of studying. And this is something that so many different companies and organizations and schedules, they say, here's the 90-day scheduling. Well, no. Because what takes you 90 days could take me 120 days. So it's not a good thing to focus on time. It's important to focus on the process and just getting better over time. This will get faster and faster and faster. And if it doesn't get faster and faster and faster, it'll get stronger in other areas. It'll get more in-depth. It'll get more critical. It'll get more educational. It'll get more whatever, right? But it's important to follow the process and get better and better over time. Any other last questions? I hope that gives you an insight into what I do. And if you need something written down, if you go to my website again, SecondChanceMCAT.com, and you look at frequently asked questions, if you click on frequently asked questions, it'll pop you to, I've embedded every answer that I've given on Quora. I'm very active on Quora.com. Quora.com is a place where you can submit a question. Anybody can submit a question, and they can hire experts, quote unquote experts, to answer them. So somebody asked, if you were to study for the MCAT again, how would you do it? And they had requested my answer, so I said, thank you for the ask to answer. Let me tell you exactly blah, blah, blah. So I've embedded them on my website. So if you have a question about studying or a strategy, you go to strategy. It'll drag you down to the part where I start talking about strategy. What's the best strategy to ace it, and you can read all these different things if you need to. Or I'm working on a blog right now, which is through Quora again, and I just called it Helpful MCAT Info. And I'm a bit trying to curate some content right now. So yeah, that's a way that you can kind of look at if you're looking, different things about stress. People ask these questions. There's a lot of sort of anxiety-inducing questions and about studying. There are other questions about studying. What's the best way to study? These different questions here. Best way to remember the 20 amino acids. So all these questions, just kind of browse through them if you have any questions on that. And that's about it. I'm on meetup.com. Should you re-drill day 1, 2, 3 on day 4, like what you already got 100% on? So this is something that it's going to take some confidence on your end. That's a great question. Personally speaking, I did not. I actually deleted the flashcards. If I got it right and I felt confident in it, if I felt like I knew it, I deleted it. Because by practicing, if it turns out 4 weeks later that I didn't know it, then it'll come back up in a different passage. It'll come back up as a concept that I didn't know that So I'll just re-add it to the bucket. But that takes a lot of confidence, I admit. It takes a lot of courage to do that, to just delete flashcards, because like you want to keep everything and you want to make sure that you don't forget anything. But I'll tell you a quick story and then we'll end it. When I was in high school, junior year or senior year, I can't remember what year I took it, I was taking an anatomy class, Human Physiology and Anatomy, HPA. And my teacher in high school, I met him after school with my father, which was, as you can imagine, very nerve-wracking for me. My dad was still doing parent-teacher conferences until I graduated high school, so yeah. But basically, I met with this teacher, and we didn't even sit down. I got into the classroom with my father. We were all just kind of standing up there, at the table. They're both very big at the time compared to me, and I'm like, my head's down. He's like, "Okay, Nicholas, what's going on?" I'm like, "Well, I don't know. Like, I'm taking all the notes. I'm doing all the work, but I don't know. I'm still getting B's and C's." And he's like, "You said you're taking notes?" I'm like, "Yeah." He's like, "Can I see your notebook?" And so I sheepishly pull out my notebook and I open it up. He's like, "Let me see, the last lecture you took notes on." So I open up the last lecture, and he's like, "Nicholas, do you know what the big chest muscle is?" I'm like, "Yeah, pectoralis major." He's like, "Why did you write it down in your notebook?" I'm like, "What do you mean? It was on the lecture. It was on the PowerPoint. I was writing down the notes." He's like, "There's your mistake, Nicholas. First of all, you didn't even complete your notes, and you didn't have time to go back and review it because you're trying to take notes on everything. If you know something, don't take notes on it. Don't write it down." Now, that shocked me, right? And that's something that I see all these students doing. So it didn't sink in until like 5 years later when I'm a senior in college. And I'm like, I'm like one of those students who, as a senior in college, I was like in the back of the lecture hall with my feet up against the chair in front of me and just like not taking notes. I'm just sitting there like staring at the professor and like asking as many questions as I can and only writing down the things that I don't know or that, make sense to me. It took me 5 years to let that sink in, but that translated into my MCAT studying, where I was very comfortable knowing when I don't know something and knowing when I do, and having the confidence to be like, okay, if I don't know it, it'll come up again, and I'll study it again. But I studied it. I got 100% on this thing. I can remove these flashcards. So what I would probably recommend you do is take it on a case-by-case basis, so that when you're done drilling, before you go back and practice, or even, when you're done drilling, you can practice, and then when you're at the organization stage, just before you drill the next time, just go ahead and again, like what we just did, go through your flashcards and delete the things that you're comfortable with. That way you don't get so swamped. So for me, I might delete more than you, or maybe you delete more than me, but the point is that, yes, you don't need to do everything 100% again and again and again, but some students do, because they're not confident. And they just want to make sure that they drill everything. And for those students, I'm very happy with that. It's okay, because do it until you get the confidence. It's just that it's going to take you a lot longer. So if you're the type of person like me who knows when they know something, okay, I know what organ stores bile, I'm going to delete that term. Then delete the term. But if you're like, I'm still kind of like, then leave it. So hopefully that was helpful. And it's 12 o'clock, so I'm gonna stop the recording right here. Thank you so much for participating, Em. I think and Sandra, I think it's very, very, very helpful for the people who are going to watch this on the recording. So I appreciate your engagement, and I will see you next week, Sunday, 10 a.m. Pacific time.